oxalase function. Which of the following enzymes converts pyruvate oxaloacetate in the mitochondria and is activated by acetyl-CoA? A. Pyruvate dehydrogenase. B. Phosphoenolpyruvate carboxykinase, PEP. C. Pyruvate carboxylase. D. Glucose 6-phosphatase. E. Fructose 1,6-bisphosphatase. Correct answer C. Pyruvate carboxylase. Explanation: Pyruvate carboxylase is a mitochondrial enzyme that catalyzes pyruvate plus CO2 plus ATP oxaloacetate, requires biotin B7, activated by acetyl-CoA, especially during fasting to drive gluconeogenesis. A equals PDH converts pyruvate acetyl-CoA, not oxaloacetate. B acts after pyruvate carboxylase. E act later in gluconeogenesis. Question 2. PEP reaction. Which of the following reactions is catalyzed by phosphoenolpyruvate carboxykinase in the cytosol? A. Oxaloacetate, phosphoenolpyruvate, B. Pyruvate oxaloacetate. C. Fructose 1,6-bisphosphate, fructose 6-phosphate. D. Lactate, pyruvate. E. Glucose 6-phosphate glucose. Correct answer. A. Oxaloacetate, phosphoenolpyruvate. Explanation: PEPK catalyzes the second irreversible step of gluconeogenesis, oxaloacetate PEP, using OSGTP. This step happens in the cytosol. B equals pyruvate carboxylase. C. Fructose 1,6-bisphosphatase. D equals lactate dehydrogenase. E equals glucose 6-phos. Question 3. Fructose 1,6-bisphosphatase regulation. Which enzyme is inhibited by fructose 2,6-bisphosphate and AM, making it a key regulatory step in gluconeogenesis? A. Glucose 6-phosphatase. B. Fructose 1,6-bisphosphatase. C. Hexokinase. D. Phosphofructokinase 1. E. Pyruvate dehydrogenase. Correct answer B. Fructose 1,6-bisphosphatase. Explanation: Fructose 1,6-bisphosphatase is inhibited by fructose 2,6-bisphosphate and AMP. Fructose 2,6-bisphosphate leads to gluconeogenesis. Enzyme is active in the fasting state when glucagon is high. A equals not regulated by these molecules. C and D equals enzymes of glycolysis, not gluconeogenesis. E part of pyruvate oxidation. Question 4. Glucose 6-phosphatase and tissue specificity. Why can't skeletal muscle perform gluconeogenesis? A. Lacks biotin-dependent enzymes. B. Does not produce oxaloacetate. C. Lacks glucose 6-phosphatase, converts pyruvate to lactate only. E only uses fatty acids for energy. Correct answer C. Lacks glucose 6-phosphatase. Explanation: muscle does not express glucose 6-phosphatase, cannot convert glucose 6-phosphate glucose. Therefore, glucose produced by gluconeogenesis cannot exit the muscle cell. A equals muscle has biotin-dependent enzymes. B equals oxaloacetate is produced in muscle for TCA. D equals not exclusive. E equals incorrect. Muscle uses glucose, ketones, and fatty acids. Question 5. Odd versus even chain fatty acids in gluconeogenesis. Which of the following molecules can be converted into glucose via gluconeogenesis? A. Acetyl-CoA from even chain fatty acids. B. Leucine. C. Lysine. D. Propionyl-CoA from odd chain fatty acids. Correct answer D. Propionyl-CoA from odd chain fatty acids. Explanation: only odd chain fatty acids yield propionyl-CoA, succinyl-CoA, enter TCA cycle. Then converted to oxaloacetate, PEP, glucose. It's odd for fats to make glucose. A. Even chain fatty acids yield only acetyl-CoA, cannot be used for gluconeogenesis. Ketogenic amino acids only also cannot make glucose.